Okay, thank you. Hi, everyone. I would like to welcome everyone for today's seminar organized from Hellenic Initiative Against Alzheimer's Disease and the graduate program in neuroscience in from the Medical School, University of Crete. It's a great pleasure and honor, actually, to have here today with us Dr. María Llorens-Martín. I will share with you a short bio for Dr. Llorens-Martín. Maria received her PhD in UCM in 2009, and after finishing her PhD on studying adult hippocampal neurogenesis and on the physical exercise effects on hippocampal neurogenesis, she joined Professor Jesus Avila's lab at the Center of Molecular Biology, Severo, Ochoa, as a postdoc fellow, where she continued her studies in adult neurogenesis, investigating the role of this unique brain process in marine models of Alzheimer's disease. In 2015, she had a postdoctoral research fellow in the University of Tsukuba in Japan. And after that, she returned back in Spain, where in 2016, she set up her independent laboratory, and she continues to work very successfully, I have to say here, in the role of adult hippocampal neurogenesis in various diseases, including Alzheimer's disease, of course. And in 2020, she was awarded a tenure research position in the Spanish Research Council. Some few words about Maria's work. She's focusing her work in adult hippocampal neurogenesis and the role of this new, actually, brain function. We know for a few years that against the Cajal dogma that the brain is is a stable organ. We know now that it can change through life, through adulthood, and we have significant evidence that adult hippocampal neurogenesis can happen, for sure in rodents, but we believe very strongly that it happens also in humans. And not only it happens, but it has also functional outcomes, especially under neurodegenerative conditions like Alzheimer's disease. Maria is very active on this field. For her work, he has rewarded as a principal investigator from recently from ERC with a consolidator grant. And she has also support from American Alzheimer's Association, the Association of Frontotemporal Degeneration, the Spanish Ministry of Economy and Competitiveness, and she has published more than 60 papers, some of these in very impressive journals like Science Journal, Nature Medicine, Nature Protocols, EMPO Journal, and others. Her work has attracted significant interest from media. I could mention here The Scientist, El País, ABC, and she's a recipient of many awards, also Young Investigator Award, the Young Female Talent Award of the Spanish Royal Academy of Sciences, and she's a full member of the Spanish Young Academy. Maria, we are very glad that we have you here, and we are looking forward to your presentation and your recent data. Thank you.
Thank you very much, Ioannis, for this wonderful introduction, and thank you very much to all the attendees. Thank you for this invitation. It's a pleasure to be with you, even though it's virtual this time, but I hope it can be in person very soon. Okay, so as Ioannis said, our work has been focused during the last decades on the process of adult neurogenesis, which can be defined as the generation of new functional neurons in the adult brain. Adult neurogenesis permits the remodeling of neuronal circuits, so it encompasses an extraordinary reserve of lifetime plasticity. As you can figure out, adult neurogenesis does not occur in all the brain regions, but it is spatially and in a time-dependent manner regulated. Adult neurogenesis, as far as we know today, only takes place in the so-called neurogenic niches, which in the brain of rodents are the subgranular zone of the hippocampus, which is the region that we have been studying so far, But also it has been demonstrated that the walls of the lateral ventricles generate new neurons that migrate towards the olfactory bulb. Here you can see a comparison of the gross anatomy of the hippocampus in two different species, which are mice and humans. In red color, you can see the dental gyrus, which is the neurogenic region of the hippocampus, which is somehow folded around the non-neurogenic regions, which are the CA1 to CA4 hippocampal subfields. Adult hippocampal neurogenesis is involved in different processes which are very important for brain function. Especially, it participates in learning and memory, and in particular in pattern separation, and it also regulates emotions and mood. Several animal models show impairments in adult neurogenesis in animal models of, for example, neurodegenerative or psychiatric disorders. So this phenomenon has attracted a lot of interest in recent decades, given its extraordinary regenerative potential. Here you can see a composition of how the life of a newly generated neuron looks like. These new neurons are generated upon the division of the neural stem cells that are resident cells in the subgranular zone of the dental gyrus, which divide and give rise to transiently amplifying progenitors and immature neurons that go through different maturation stages before becoming fully mature and fully synaptically integrated into the circuitry. The classical ways in which this phenomenon has been studied are the use of specific cell markers that are transiently expressed during discrete periods of this maturation process. In this way, we can combine the expression of different markers and classify the cells that are at a specific maturation stage during adult neurogenesis. Adult neurogenesis has been demonstrated to occur in rodents since 1962, and nowadays no one questions the occurrence of this phenomenon in the brain of rodents. Regarding the occurrence of adult neurogenesis in the human brain, this is a summary of the literature that has been published so far, and in green color you can see all the papers that support the occurrence of adult neurogenesis. But in gray color, I have represented some manuscripts that deny or question the occurrence of this phenomenon. So in the light of these discrepancies, in the light of the fact that different labs using apparently similar methodologies came to totally opposite conclusions, we wondered whether slight methodological differences could lay behind these discrepancies. This is a very simple summary of the pathways that one can take to process a human sample. Something that looks very simple is actually very complicated, and some of these pathways, which indicates the choice of a different methodological path, are incompatible with others. So in our experience, what we have found so far is that each lab has its own methodology, and in the end, the quality or the characteristics of the samples of different labs are totally different. So we could not study the impact of each of these stages on the final results, but we focused our attention on determining whether fixation, which was very important in our mouse studies, could be really impactful for our capacity to detect markers of adult neurogenesis in the human brain. So to answer this very simple question, what we did was to obtain the whole hippocampus from several subjects and divide these hippocampi into different fragments. Next, what we did was to fix each of these fragments obtained from the same subject during a different period of time. So one would expect that the number of cells that could be detected should be the same because all the fragments come from the very same subject. So in order to test this hypothesis, what we did was to count the number of double cortin positive cells, which is, I would say, a universally used marker of immature neurons. And to our surprise, what we saw was that with very short fixation times, shorter than 12 hours, we observed a beautiful staining with double cortin that labeled immature neurons, very similar to those that have been described in rodents. But what was really surprising for us was the fact that after 12 more hours of fixation, a sample obtained from the same subject showed a totally different aspect. Here you can see how a sample fixed for 24 hours displayed this high intensity background and a lack of specificity in the signal. So we were almost unable to detect positive cells in samples that had been fixed for 24 hours. And what is most important in this case is the fact that most of the samples in most of the brain banks worldwide used to fix human brain samples for several weeks or several months. So at that time, we started to work on a protocol that allowed us to recover the signal that had been lost due to this excessive fixation with aldehydes. Unfortunately, we described this protocol, which is nothing special, but the combination of 3 steps that resulted very effective. As you can see here, in samples fixed for 24 hours that were subjected to sodium borohydrate to reduced aldehydes, a special, very mild protocol of antigen retrieval and autofluorescence elimination, we could improve the signal with double cortin, and not only double cortin, but many other markers of adult neurogenesis in the human brain. So the conclusion of these experiments was the fact that fixation is important for us to be able to detect the cells that in fact are there. And if we take a closer look at the literature, we can see that the 3 papers that denied the occurrence of adult neurogenesis used much longer fixation times. Moreover, 2 recent papers that also questioned the occurrence of adult neurogenesis did use short fixation times, but used a harsh antigen retrieval protocol, which in our hands makes that antibodies detect in a specific signal. So these 2 papers detected double cortin positive cells in the dentate gyrus, but due to the conditions of the antigen retrieval that they used, likely detected double cortin positive cells in other brain regions. So these 2 papers from our lab demonstrate that fixation critically impacts our capacity to detect markers of adult neurogenesis in the human brain. So the cells can be there, but we may not be able to visualize them. So in order to biologically validate the signal that we detected with anti-double cortin and other markers of adult neurogenesis antibodies, we made sure that these signals were only observed in the dentate gyrus, but not in other non-neurogenic regions. Moreover, the aspect of these cells, as you can see here, is the classical morphology of a granule neuron with this very thin cytoplasm, lack of nuclear staining, and also weak staining in the apical dendrites. Moreover, double cortin is not expressed in glial cells, such as astrocytes or microglia, and it cannot be detected in blood vessels, demonstrating the specificity of both anti-double cortin and other markers of adult neurogenesis antibodies to detect the proteins of interest. So in order to make a first exploration of adult neurogenesis from a quantitative point of view, we studied this cohort of 13 neurologically healthy control subjects, which were not only neurologically healthy, but also neuropathologically controlled. In this case, we followed the criteria that they were at Bragg tau stage 0, so no tau phosphorylation in different brain regions. And in collaboration with Dr. Alberto Rábano, we observed that the postmortem delay up to 38 hours did not make a big difference in the number of double cortin positive cells that were detected. Also, the gender did not appear to have a strong effect. But in contrast, the age of the subjects, as have been described in rodents and also suggested to occur in the human brain, have a negative effect on the number of immature neurons. So this data means that the rate of adult neurogenesis might be decreased during physiological aging. And this is not surprising. We know this for several decades in rodents, and it is somehow expected that the same occurs in humans. However, not all the double cortin positive cells are similar, but we observe marked differences in the morphology and the position of these cells within the granule cell layer. This data indicates or suggests that these cells can be at different maturation times. Unfortunately, we cannot use thymidine analogs, for example, to birth date these cells, so we cannot know how old they are actually, but we can make an indirect quantification. So what we did was to colocalize the expression of double cortin with that of other markers that are expressed during shorter periods of time. So we could try to make a parallelism between the expression of these markers and the morphological properties of these cells. So as you can see here, double cortin colocalizes with markers of distinct stages of adult neurogenesis, including proliferative neuroblasts and proliferative cells, but also immature neuroblasts, such as those that co-express calretinin, are more differentiated immature neurons, those that express calvindin. So we selected these two apparently extreme populations of immature neurons and compared their morphology and their positioning in the granule cell layer. You can see at first sight how these cells, these populations of cells, are quite different. So calretinin positive cells are small, horizontal, and have several primary apical neurites. But those that are at most more advanced stages of differentiation, such as those that express calvindin, have only a single primary apical dendrite which is oriented vertically towards the molecular layer. So this data indicates that adult neurogenesis seems to be a dynamic process in humans as it is in all the species in which it has been studied. But something that was somehow criticized about our initial work was the putative absence of neural stem cells. And it is true that at that time, in 2019, when we attempted to make an immunohistochemistry against nestin, which is one of the best known markers of neural stem cells, this is what we obtained: no marker, no specific signal, and a lot of background. So what was the problem at that time? We were using, as most people do, a standard detergent which was Triton X100. And that detergent is too strong for the antigen nestin to be detected by antibodies. So here you can see what a tremendous change when Triton is replaced by saponin, which is a milder detergent. You can see how by using saponin in the same protocol, we can detect different markers of neural stem cells, such as nestin, SOX2, vimentin, and of course GFAP. Moreover, I would like you to keep in mind this morphology of these cells because it will be important. So, anyone can question whether these cells are actually neural stem cells, because all these markers can also be expressed by astrocytes. So, to the best of our knowledge, the best criterion that has been used in the literature to identify neural stem cells is the expression of all these markers and the absence of the astrocytic marker S100 beta. In fact, when you stain a human tissue with an anti-nestin antibody, you can label two different populations of cells that are clearly represented here. On the one hand, you can see these small astrocytes, which are nestin positive, but also S100 beta positive. And on the other hand, you can see these cells, which have a totally different morphology with these long processes that traverse the granule cell layer and ramify in the molecular layer. These cells are S100 beta negative. So these are the quantifications that demonstrates that the morphology of nestin positive, S100 beta positive, and S100 beta negative cells are completely different. demonstrating or pointing to the existence of actually neural stem cells in the human dentate gyrus throughout lifetime. These cells do not have S100 beta expression, are located in the subgranular zone, totally opposite to these astrocytes, and have these characteristic long apical processes. Moreover, one would expect that these cells give rise to transiently amplifying progenitors, and in fact you can see here how these cells are dividing in the subgranular zone, which are detected with different proliferation markers. But one could say that these cells are astrocytes, so we wanted to have a better idea about the phenotype of these proliferative cells that were detected. So as you can see here, 90% of the proliferative cells are double cortin positive and are located in the subgranular zone. So this data suggests that the most of the proliferative cells that can be detected in the adult human dentate gyrus are neuroblasts that are located in the subgranular zone. So together with the previous evidences that are available in the literature, our data demonstrating the presence of immature neurons, stem cells, and proliferative cells in the human dentate gyrus supports the notion that adult neurogenesis is a robust phenomenon in the human dentate gyrus until the 9th decade of life. However, what we have been seeing so far is people which are neurologically healthy. So what happens during both pathological and physiological aging? And in this case, it is important to highlight the impact that the niche in which these neurons grow has on the regulation of adult neurogenesis. You can see this very simple scheme in which immature neurons and neural stem cells are accompanied by the presence of an extensive network of vascular elements, astrocytes, and microglial cells. So all these elements provide the trophic support necessary for these cells to survive and grow in the dentate gyrus. What we have been trying to find out is which are the potential causes of this decrease in the number of immature neurons that we observe during physiological aging. And surprisingly, we didn't find any vascular changes or in the presence of astrocytes. But microglial phagocytosis is slightly different between young and aged subjects. So here you can see how microglial cells emit these phagocytic pouches, which are structures that emerge from the cytoplasm of these cells and allow them to phagocytose these dysfunctional or apoptotic cells that are present. And in fact, almost 40% of the apoptotic cells that are present in the dentate gyrus are phagocytosed by microglial cells. What we have been studying in mice and think that we are able to apply to the human brain is the fact that the morphology of the microglial cells reflect their function and their phagocytic capacity. So you can see how the presence of these phagocytic pouches correlates with morphological differences in microglial cells. And the higher the number of phagocytic pouches the microglia has, the less is the number of pycnotic cells that are not being phagocytosed. So, even though the number of microglial cells do not change with age, what we saw is the number of phagocytic pouches that are expressed by these cells is reduced during physiological aging. So this data talks about a possible impairment in the functionality of microglial cells throughout aging that has been also described in rodents. And we know from mice studies that this decrease in the functionality, in the capacity to phagocytose these pycnotic cells by microglial cells are aggravated by the course of distinct neurodegenerative diseases. So we wondered what happened both with adult neurogenesis and the homeostasis of the dentate gyrus neurogenic niche in patients with distinct neurodegenerative diseases. So the first disease that we studied, because it's highly prevalent, it's in fact the most common type of dementia in industrialized countries, was Alzheimer's disease. As you know very well, this disease is characterized by the loss of neurons in specifically sensitive brain regions, and also by lack of synapses in the same. So we collected during almost 10 years a collection of samples that had been processed in the way I told you before, 24 hours of fixation and short postmortem delays, in collaboration again with Dr. Alberto Rábano. And we collected this group of patients that were at distinct Brach-Tau stages of the disease. As you know, the Brach-Tau stage 1 is the first disease stage, which goes progressively towards Brach-Tau stage 6. So the first thing we did in these patients was to quantify the number of immature neurons that could be detected in their brains. And surprisingly, we detected a 33% decrease in the number of immature neurons in patients at Brach-Tau stage 1. As you also know, these stages of the disease are very early, and most of the time they are silent, so they are prodromic stages of the disease. So even before the deposition of amyloid beta and phosphorylated tau in the dentate gyrus, which occurs around Brach-Tau stage 4, we detect a decrease in the number of immature neurons. Of course, this number is further reduced during the disease progression, but it is highly remarkable that cells are being impaired very early in the disease. In fact, results from the group of Orly-Lazarov demonstrated that patients with mild cognitive impairment already show a decrease in the number of proliferative neuroblasts. This decrease is specific for immature neurons. As you know, it is very well characterized that the total number of mature neurons in the dentate gyrus is not modified during the disease progression. But as we detected a reduction in the percentage of double quartile positive cells that expressed terminal differentiation markers such as calvindin, what we concluded was that the number of cells is reduced, but the differentiation of these cells is further impaired. So it can be that Alzheimer's disease is blocking the maturation of these processes, of these cells at different time points, but also it is possible that Alzheimer's disease is impairing the homeostasis of the dentate gyrus neurogenic base. This is something that we are currently studying, and we have, for example, found out that the proliferation of progenitors is decreased in these patients. So next, we wondered whether this was a peculiar situation related to the fact that Alzheimer's disease directly targets the hippocampus and the dentate gyrus, but what happened with other diseases in which the dentate gyrus or the hippocampus was not the main target. We wanted to find out what happened with adult neurogenesis in other diseases, such as amyotrophic lateral sclerosis, ALS, Huntington's disease, Parkinson's disease, dementia with Lewy bodies, and frontotemporal dementia. These diseases are all more or less prevalent in the old population, but frontotemporal dementia is also frequent in subjects below 65 years of age. So what do these diseases have in common? Nothing, apparently, just neuroinflammation and neurodegeneration and cell death. But we didn't know whether what we observed in patients with Alzheimer's disease was also going to be observed in these patients, or we could have the opposite situation. So in this case, we have the opportunity to study the whole process of adult neurogenesis, starting with the presence of neural stem cells. And to our surprise, what we observed was the fact that in patients with ALS, Huntington's disease, and Parkinson's disease, we found an increase in the number of nesting positive S100 beta negative cells, which means that the population of neural stem cells seem to be increased in these patients, which is different from the situation we observe in patients with dementia with Lewy body and frontotemporal dementia. So one would expect, or not, that the whole process of adult neurogenesis goes in parallel. So what would happen, or what would we expect regarding proliferation of these cells? well, regardless what we expected, proliferation did not change in patients with ALS or Huntington's disease. It was increased in Parkinson's disease and not modified in the other diseases. In fact, the number of proliferative neuroblasts that have exited the cell cycle was reduced in patients with frontotemporal dementia. So if we compare the increase or no modification in the number of neural stem cells with the variations in the number of proliferative progenitors, we could conclude that the balance between the quiescence and activation of these cells seem to be disbalanced or seem to be impaired. Increase in the case of patients with Parkinson's disease or unchanged in the case of patients with dementia with Lewy body. So apart from this complicated puzzle of data, what I want to show you here is the fact that it seems that each population of cells has a different vulnerability to each of these diseases. In fact, when we studied immature neurons labeled with double cortin, this number of cells was increased in the same 3 diseases and unchanged in the 2 that are on the bottom. So together with the increase in the number of radial glia-like cells, does this mean that the process of adult neurogenesis is increased in neurodegenerative diseases? One could think so, but the problem is that the morphology of these cells is strongly altered in all these neurodegenerative diseases. Not all the stages of differentiation are equally altered, but the overall conclusion is that the morphology of these cells cannot be developed properly. So you can see how many of these cells display several primary apical dendrites or are mislocalized in the subgranular zone, which demonstrates or suggests that the maturation of these neurons is inappropriate. So what happens with these neurons? They are there in an increased number. Do they die? In fact, this is what we could conclude from the data. In all the diseases, there is an increased density of apoptotic cells in the dentate gyrus. So even these areas are so far from the main areas that are affected by these diseases, for example, in ALS, there is an increased number of apoptotic cells here. And we know from mouse studies that the main cells that undergo apoptosis are those that come from adult neurogenesis and not the mature neurons that have been developmentally generated. Of course, we don't know if this is the case in humans, but this increased number of immature neurons is not reflected in an increased number of total mature neurons, which is not changed in any of these diseases. So we thought that these impairments in adult neurogenesis could reflect also alterations in the homeostasis of the dentate gyrus neurogenic niche. So we didn't find increases or decreases in the number of microglial cells, but in all these diseases, the phagocytic capacity of microglial cells was decreased. So we have an increased number of apoptotic cells, a higher number of immature neurons that are not maturing correctly, but the microglia might be exhausted. We know that this situation occurs, for example, in patients with Alzheimer's disease, and we believe this can be the same case for patients with these other diseases. In fact, there is a generalized astrogliosis in these patients. The number of S100 beta astrocytes is quite increased. And also, surprisingly, there is an increase in the vascularization of this neurogenic niche, which can be, well, it can be positive because it increases the trophic support of the newly generated neurons, but it can also amplify the neurodegenerative environment produced in distal brain regions. So all these data suggest that the global homeostasis or the equilibrium of this neurogenic niche is impaired in all the neurodegenerative diseases studied. However, when we look at the general picture, we see an increased apoptosis in all the diseases and impairment in this neurogenic niche, but there might be something that is specific for each of these disorders, given that all the cell subpopulations that compose the process of adult neurogenesis are not equally affected by these diseases. you can see here a summary. It's also in the paper, showing how different aspects, such as the balance between proliferation and quiescence of the neural stem cells, or the morphology of immature neurons, is differentially sensitive to the appearance of these disorders. So, just as a summary of what I presented you today, our work and other evidence in the literature demonstrate the presence of neural stem cells, proliferative cells, and immature neurons in the human dentate gyrus. We have also described the existence and the potentially main components of the human dentate gyrus neurogenic niche. We have, I consider, demonstrated that neurodegenerative diseases impair in an apparently unique way adult neurogenesis and the equilibrium of the neurogenic niche in humans. And with the support of our recently awarded ERC Consolidator grant, what we are currently investigating are the molecular and cellular triggers that can control adult neurogenesis in humans and that can lead to this specific dysregulation in each of these disorders. So I would like to introduce the most important part of this seminar, which is the composition of my group and these wonderful young researchers. I would like to thank our collaborators for all their support during the last years, and our sponsors. So thank you very much for your attention, and I will be happy to answer your questions.
Thank you very much, Maria. Exciting and very intriguing presentation, huh? So the discussion is open for questions. If someone has something to ask, okay, I can see a question on chat from Eugenia. Thank you for a great talk. What is your take regarding the recent single nucleus transcriptome study from the CESTAN group showing absence of cell type specificity for double cortin?
Well, I think they are very interesting data that demonstrates quantitative data that demonstrates that the number of cells that need to be analyzed to detect some populations of cells, such as neural stem cells, is really high. And if you don't start with an adequate or sufficient number of cells, you will not detect proliferation or radial glia-like cell clusters. So, of course, that data has been published, but there are other data that has been published in which the presence of proliferative clusters with neurogenic potential are identified by single-cell RNA-seq. So I think, of course, the story of human adult hippocampal neurogenesis is not over, and there are many interesting data coming from different labs that hopefully will be published soon and will help to disentangle this controversy regarding the single-cell RNA-seq studies, which do not agree.
Okay, thank you. Any other question?
Yannick, can I do a question, make a question?
Yes, of course.
Okay. Hi, Maria. Thank you very much for this wonderful presentation. It's very exciting. And so how can we increase the adult neurogenesis? We know that there are some studies saying about exercise, that they can increase this. Can you tell us a little bit more about this, or if we know the molecular mechanisms behind this?
Yes, in mice, we know that there are different pathways that leads to increased adult neurogenesis. Physical exercise is one of the cheapest one, probably. Mm-hmm. We know that environmental enrichment and also social enrichment are healthy ways to increase adult neurogenesis in mice. Of course, we don't know whether this is the case in humans, but it would be interesting to explore at least. And regarding the molecular mechanisms, we know that adult neurogenesis is regulated by plenty of factors, mainly growth factors, BDNF or IGF-1 or PEGF are maybe some of the most, or of the well, the better ones, the ones that have been more studied, but there are many factors that increase adult neurogenesis, and also that decrease it, like different soluble neuroinflammatory factors.
Thank you.
Thank you, Vasiliki.
Dimitra Thomaidou has raised her hand. So Dimitra, please go to your question.
Thank you very much, Maria, for the very nice, exciting talk. I have a question to make. So, what your results show decreased numbers of double coating positive newborn neurons in neurodegenerative diseases. Do you think that the only reason for this is cell death, because you see no change in proliferation rate, or it can be that newly produced neural stem cells do not progress to the next maturation stage, so they don't also give rise to double coating positive neurons? Could be this an alternative mechanism? Or
yeah, I'm sorry. Sorry. Yeah, I absolutely agree. I think there are many mechanisms that could explain, and several of them could be taking place. So, for example, the time during which double coating is being expressed can also be changed. The process can be slower or faster. Double coating expression can start earlier or later. So, in addition to the lack of adequate proliferative capacity of neural stem cells, there are many factors. I'm sorry if I said that is the only explanation. I don't think it's the only explanation. I think there are many factors that could explain what we observe.
Have you seen any? Have you compared, for example, BRDU positive double coating cells to BRDU negative, let's say, double coating cells, to see if there is an increase in the population with neurodegenerative diseases of BRDU?
Yeah.
In relation to BRDU incorporation.
Yeah, unfortunately, BRDU, as far as we know, it cannot be used in humans.
Yeah.
But in mice, for example, in one of the mouse models that we have studied in the lab, which is a GSK3 overexpressed in mice, we have made the time course of double coating expression in BRDU positive cells. And in this case, what we found was a lengthening of the period of double coating expression. But these animals have more double coating positive neurons. So maybe in the case of humans, it occurs the opposite, that double coating is expressed during a shorter period of time, or the switch on of double coating expression is delayed. So finally, the period of time in which you observe the expression of the marker is shorter.
Thank you very much.
Thank you.
Okay. Yannis Agana, you are the next one. Let's go on.
Thank you very much for this very, very exciting talk. I was wondering if you have any evidence that, given that many of these neurodegenerative disorders have a genetic basis, like the presennin 1, 2, and APP mutants in Alzheimer's disease. Of course, the ApoE susceptibility gene in Alzheimer's disease, and also variants in the C9orf72 or the TDP43 gene in frontotemporal dementia. So, do you have any evidence that these pathogenic variants in these genes affect the way neurogenesis is executed in the hippocampus?
Yeah, we would be very interested in studying familial cases of Alzheimer's disease, but unfortunately, none of the cases that we have samples from have a familial origin. And this is something that we have investigated with the brain bank because it would be very interesting to know. But none of them have a familial or a known genetic origin. And in the case of the other diseases, what we did was a very broad representation of the amount of phosphorylated TDP43 or phosphorylated tau or precipitated huntingtin in the dental gyrus. And in none of the cases, the presence of immature neurons correlates with the presence of those molecules. So we think it might be some, I don't know, some long-distance signaling mechanism. And if you see in the epidemiologic data of our patients, I think they are quite well classified regarding the presence of aggregates of TDP43 or, as I said, Huntington, etc. But I think the numbers of patients we have are not sufficient to really affirm whether this is an effect derived of whether these patients are TDP43 positive or not. So I would like to have more patients to make that comparison.
If I may follow up on this, it's in our brain bank also that we find a lot of Alzheimer's patients with TDP43 deposits. So maybe this is a common theme behind this. But I was talking also about the ApoE status. So in Alzheimer's brain, you have several E4 carriers. So do you have any difference between having the E4 allele or not having the E4 allele?
We haven't studied that, but I think we can dig into the ApoE4 phenotype or genotype of these patients. So yeah, thank you for the suggestion. I will let you know if we have some interesting patients among them, which I'm not sure that they are ApoE4. I'm not sure. Thank you.
Okay. Thank you, Yanni. Konstantinos Ioannou has raised his hand, so Konstantine, please go on.
Hello, Maria. Thank you for the nice talk.
Hi,
Actually, I have two questions. The first one is that do you have observed any correlation with the education level of the individual and the percentage about of the impairment of these cells with age? And the second question is, as you said, in Alzheimer's disease, these cells are already impaired before BRAC stage 3 and 4, that are usually the stages that we detect clinically Alzheimer's disease, if we are lucky, and also are the stages that we start to see neurodegeneration. So if these cells are already damaged before the stages, what creates this impairment? Maybe has amyloid beta a role about that, because it's supposed that it precedes tau accumulation. Mm-hmm. But I didn't see any data about beta amyloid in these individuals. That's why I asked.
Yeah, so thank you for your questions. I would really love to correlate lifestyle habits or education level and adult neurogenesis rates. But unfortunately, of this cohort of patients, we don't have enough information from all of them. So the numbers of the patients from which we have some information are not enough for us to make an assertion. And regarding the presence of amyloid beta, what we have quantified is the deposition of amyloid beta and phosphorylated tau in the dentate gyrus. And we find a correlation, even though the accumulation is not statistically significant at BRAC tau stage 1 and 2, we do observe a correlation between the number of immature neurons and the position of amyloid beta and tau. But it could be that the presence of oligomers, both from amyloid beta and tau, which cannot be detected by standard antibodies, is occurring earlier. And it's not us, but there is a group of young from the US who they suggest that maybe this impairment in adult neurogenesis can trigger neurodegeneration in layer 2 of the entorhinal cortex through denervation of the axons. So I don't know, I don't think we have enough data to affirm that, but it's a very interesting hypothesis. It's very strong to say that adult neurogenesis impairments can be a cause for anything in Alzheimer's disease patients, but at least it could be related to the massive neurodegeneration that is observed in the entorhinal cortex. But I don't know anything about the mechanisms. It could be also neuroinflammation that is, we know that is very much affected several decades before the appearance of cognitive impairment. So it could be.
That's true. Yeah, thank you.
Thank you.
Okay, thank you, Konstantina. Nice to see you. Any other question?
Yanni, there is a question in the chat from Paul Lucasen.
Okay. Yes.
Yeah.
Hi, Dr. Lucassen. Nice to have you here today.
Hello. Nice to meet you all, and thanks, Maria. Very good to see you. It's a brilliant talk as ever. Fantastic work. Very nice to find this opportunity online to listen to your work without having to travel. I was just wondering. Well, the question is already in the chat. You showed a very nice relation between apoptosis and microglia. I was just wondering. In terms of rodent work, we know that apoptotic cells are only around for a very short period of time, right? So if you are able to trap an apoptotic cell in the process of dying in a very thin tissue section of a brain that has been sick for several years, it's a lucky shot. And usually in rodents, it takes over 72 hours that such an apoptotic cell is present, and then it is being phagocytosed by microglia. So I'm always surprised if people see so many apoptotic cells in a brain. Do you think it is related to the microglia, that they are just slower and that therefore more apoptotic cells are visible, or do you think there's another process that explains their presence so clearly in your material?
Well, we have been very surprised because I honestly didn't know very well the numbers from the rodent mice. And what we have seen is that the numbers are quite similar to those observed in rodents. For example, we observed that around 40% of the apoptotic cells are surrounded by microglial cells, which is almost identical to the numbers that Amanda Sierra, for example, observed in rodents. Also, the number of phagocytic pouches in the microglial cells is very similar between rodents and humans. And I don't know, I think 50 microns of a huge dentajar sample is a lot of tissue. So I don't think the density of pycnotic cells that we observe are so high. For example, compared to the number of immature neurons, it's 20% more or less. So I don't know. I would not say there are very high numbers.
No. And if they would change the ratio between the newborn and the dying cells, would you not also assume that maybe the dentate as a whole either grows or becomes smaller? And did you take that into account in your measurements? Or will you be doing that? Because in Alzheimer's, it's surprising that you don't see that much changes in the dentate. I'm always surprised by that. You see losses in the CA1 where cells really die, and that area is much smaller in Alzheimer's, but in the dentate, it always stays more or less the same size. And if you find this reduction in newborn cells, you would maybe assume that the dentate becomes smaller, but that's also not the case. So what's your view?
Yeah, and that reminds me of very interesting data that we obtained in control subjects, which is that during aging, both the number of immature neurons and the number of apoptotic cells is decreased during aging. So it seems that the whole process can be slowed down or negatively regulated. And I think it's something that also occurs in rodents, that both things occurs in parallel. And I honestly, I admit that I was very surprised to compare the human data with the rodents in terms of numbers, because I would have expected an increased number of apoptotic cells in aged subjects, but it's totally the opposite. We found a reduction.
Yeah, that's always surprising.
Yeah.
And my last comment, and you talked about environmental factors and lifestyle factors. Is there any role that you see for stress or stress hormones in these patients that might have interfered with your data, because it is such a strong suppressor of neurogenesis in rodent work? It must be relevant at some point in humans as well. Is there any way to measure it, or do you plan to do that, or?
Well, I think we haven't considered stress specifically, but we would like to compare soluble factors in blood from these patients with the levels of adult neurogenesis. And I think those samples might be available. I'm not sure whether from all the subjects or not, but from many of them could be available. And I think in the light of your work, it would be really fascinating to look at the stress and the impact on adult neurogenesis. We are also studying patients with psychiatric disorders, in which I think we can have a better idea of the role of stress.
Yeah. Okay. Well, thanks very much for your answers, and again for your very nice talk.
Thank you, Paul. Thank you very much.
Thank you. Any other question? Perhaps I should do last one. So because we are speaking also about psychiatric disorders and the role of antidepressants, for example, in adult neurogenesis, do you have any evidence in your human samples, Maria, about neurotrophin receptor expression or neurotrophins per se? Because there is also a debate about which neurotrophin receptor is expressed in which stage, in which cell. Actually, we know that BDNF is increased after exercise, the science paper from Rudy Tanzi and Fred Gates, but I haven't seen any data for neurotrophin receptor in human samples, actually.
I'm looking in my literature base. We don't have our own data about it. I think we have tried staining with some TrkB receptor with not much success, I think. But the only work I know is that from Maura Boldrini, who showed that adult neurogenesis is decreased in patients with major depression, and I think that they showed increase in neurogenesis after antidepressant treatment. I'm not sure about that, but or at least differences in the responders versus the non-responders to the treatments. And I think that could be related to the expression of the neurotrophin receptors, but we haven't tested on our samples.
Yes, that's right. Because there is this discussion, this nice work from Eero Kastren about the connection of the depressants at TrkB, and through this way, the upregulation of adult neurogenesis. So it would be nice to see in these human samples, and especially in AD patients, what's happened.
Yeah, absolutely. It will be a very interesting interaction.
Okay, I see in chat. Thanks, Maria, for nice talk. Is there any evidence on the effects of nutrients like glucose on neurogenesis?
Well, on mice, there are a lot of studies examining the effects of diet in general, and in general, to make this story short, caloric restriction increases adult neurogenesis, and then there are lots of work, not directly on glucose, but on diabetes animal models and insulin and IGF-1 signaling factors, for example. But I don't know if glucose in particular, I'm not sure. I don't think about glucose in particular. I don't remember. Maybe Paul remembers any study on, or Sihon. I don't know. I don't remember any specific work.
Okay, thank you. Any other question from the audience?
One more from.
One more, Kasatina, come on.
Have you maybe checked the GFAP from astrocytes, because now it's becoming a very emerging biomarker in the field of Alzheimer's disease, and so there are more and more papers about its role and about that it can detect much earlier some changes.
Well, we have stained with GFAP, and we observe some similarities with S100 beta staining, but also some differential regulation in some of these diseases. I don't remember exactly the data, but I know that in some diseases we observe increase or not increase with GFAP and the opposite with S100 beta, but not in Alzheimer's disease patients, not yet.
You haven't done this yet?
Not in Alzheimer's disease, sorry, it was in the other neurodegenerative diseases, because it was in the recent paper.
Okay, because it seems that the plasma levels, for example, in GFAP can differentiate Alzheimer's for other types of dementia, so maybe it's interesting.
Yeah, it's interesting to see. We are near to do that, but we haven't done yet in Alzheimer's disease patients.
Thank you.
Great. So if there is no other question, thank you again, Maria, for your talk, for your presence here today.
Thank you very much.
As Paul said, till next time.
Till next time, in person somewhere.
Yes.
Thank you very much, Maria.
Thank you all.
Thank you very
Thank
much.
you all for
Thank you on behalf of HIAAD as well. Bye.
Thank you. Bye.
Bye.